Welcome to this sneak peek into some lab experiments performed by students in modules offered in the degrees of BSc Biomed and BSc Biotechnology Management at Pearson. We start with molecular biology. DNA profiling or DNA fingerprinting is used to solve crimes, in medical diagnostics, and to confirm relationships like paternity. Genetically, we are 99.5% similar to each other. However, DNA fingerprinting is a modern technology that analyzes that remaining 0.5% difference from the non-coding regions of each individual's DNA. It uses samples from skin cells, blood, saliva, hair root, or even bone. And when it amplifies the samples through polymerase chain reaction and runs them on agarose gel electrophoresis for analysis. Plant propagation. Did you know that people have been cloning plants in one way or another for thousands of years? For example, when you take a leaf cutting from a plant and grow it into a new plant, you are cloning the original plant. This is because the new plant has the same genetic makeup as the donor plant. In this experiment, students used different plant tissue culture media combinations to establish plant cell cultures from mustard leaf explants. The ultimate aim is to generate whole clone plants. Histology. How do pathologists determine whether someone has cancer, and specifically what form of cancer it is? In this practical, students dissect a tissue sample and expose it to the chemicals for tissue processing. You end up with a wax-covered sample that can be sliced thinly using a microtome and exposed to dyes to visualize the cell under the microscope, just like a pathologist does in their practice. Industrial and environmental biotechnology. The increase in environmental pollution due to industrial activities necessitates the need for more environmentally friendly ways to clean the environment. You will in this practical search for bacteria, fungi, and plants to clean up oil spills, acid mine drainages, and wastewater. You'll search for these organisms from different environments and test their bioremediating capabilities and identify them using both morphological and molecular characterization techniques. Ethnobotany. Do you enjoy herbal detergents or products just because of how they smell? Well, there's another reason why these ingredients are added to the products. It's to enhance their antimicrobial and antibacterial activities. In this experiment, students investigate the activity of medicinal plants against selected microorganisms. They do so by using a microdilution broth method, which involves mixing different concentrations of plant extract and bacteria. Following subsequent incubation, microbial growth is then determined by use of growth indicators. Agricultural biotechnology. Genetically modified organisms are such a controversial topic in the agricultural sector and others, with so many people debating pros and cons. In this lab session, we use molecular techniques involving DNA isolation, polymerase chain reaction, and gel electrophoresis to distinguish between genetically modified maize and those that are not genetically modified by looking for some specific DNA sequences that can only be found in genetically modified maize. Food and water microbiology. Imagine the following scenario. A group of individuals fall ill after a dinner function, and you need to investigate this foodborne disease outbreak. You begin by evaluating the disease symptoms in order to narrow down the probable causative agent. This is then followed by a detailed analysis of all implicated food samples, which leads to an identification of the causative agent. Your task is also to advise on how the agent has entered the implicated food source and measures that can be employed to prevent or minimize the likelihood of a reoccurrence of such an outbreak. Nutraceuticals and functional foods. Vitamin C is an important vitamin that's essential to maintain your health and specifically your immune system. Various products such as MedLemon and Everflu C contain vitamin C, also known as ascorbic acid. In this practical, students determine whether the claimed amount of ascorbic acid in medicines, supplements, vegetables, and fruits is what is actually present in the item. Just a warning for the next slide. If you are squeamish because of blood or tissue or organs, please look away now. Anatomy and physiology. Your body's ability to keep you active and healthy relies on a functional cardiovascular system made up of the heart and blood vessels. In order to understand how the parts of this organ system function so flawlessly, students perform a dissection of a bovine heart. They inspect the macroanatomy of the heart and identify various important components to better understand the organ's function. And lastly, food technology. Yogurt is a food that is enjoyed by both the young and the old and is produced by bacterial fermentation of milk. Modern yogurt production involves culturing milk with live lactic acid-producing bacteria, which are also beneficial probiotics, making yogurt a healthy food snack in addition to its richness in calcium. The lactic acid coagulates the milk products, making yogurt thick and slightly sour in flavor. In this experiment, students make yogurt using this process and assess the effects of temperature and stabilizers on the texture and the taste of the yogurt. And yes, the students get to eat the yogurt too. So we finish off the snippet with the words of William Blake: "The true method of knowledge is experiment."